Hello students, this is the lesson video for lesson 2.20 and we'll be going over our next macromolecule, nucleic acids. Here's a general overview of what we'll be going over today. We'll be going over the macromolecule nucleic acids. The two examples we'll be looking at is RNA and DNA. We'll be looking at its structure and how it's made up of 5 carbon sugars, deoxyribose and ribose, phosphate groups, and nitrogenous bases. The goals for this lesson is that you'll be able to recognize that nucleic acids are macromolecules with information on how to build and maintain an organism. You'll be able to understand the roles that DNA and RNA play in the body, and you'll be able to understand the structural similarities and differences between the two different types of nucleic acids, DNA and RNA. And lastly, remember our overarching goal, which is to describe the role of carbohydrates, proteins, lipids, nucleic acids, and water in living things, and we'll be focusing on nucleic acids. Nucleic acids are the last macromolecules that we'll be talking about, and their main function is that they store and implement genetic information. So whether you're looking at a tall redwood or a small plankton in the ocean, all living things contain nucleic acids. And the reason why all living things need nucleic acids is because every living thing needs instructions for how to grow and develop. So deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA, are nucleic acids. Those are the two different types that we'll be talking about today. DNA contains an organism's genetic information that it's passed on from generation to generation. It's the permanent copy of an organism's genes. Whereas RNA uses the instructions from DNA and allows other parts of the cells to get access on how to build proteins. So you will learn many more details about how nucleic acids store and translate information in another lesson, but we're going to be focusing in this lesson about the similarities and differences of RNA and DNA. DNA is shaped like a twisted ladder or a double helix. So it was actually pretty difficult to figure out what the actual structure of DNA, but in 1953, James Watson and Francis Crick were able to determine that DNA was arranged in a shape called a double helix. So what exactly is a double helix? So you can imagine a ladder like you see right here, and if you twist the end of the ladder in a different direction, you'll get this structure right here, which we call the double helix. You can also think of it a different way. You know what a slinky toy is? A slinky is actually a helix, and it spirals around and around. So if you imagine, if you have two slinkies running parallel to each other, so side by side, they would make a double helix. You will soon see that one kind of RNA is shaped like a single helix, so you can say that it's shaped like one slinky instead of the two that make up DNA. So again here, this is what a double helix looks like. If you were to untwist it, it would look like a ladder. And if you were to look at just one strand of the DNA, you'll see that each of the strands actually have a series of nucleotides, which are the building blocks of nucleic acids. So nucleotides are again the repeating units of nucleic acids, and they're made up of 5 carbon sugars, a phosphate group, and nitrogenous bases. A nucleotide is a small molecule made up of those 3 things, and as you can see here, the phosphate group is made up of phosphorus, which is right here, and 4 oxygen molecules. And as the name suggests, the nitrogenous base contains nitrogen. So the nitrogenous base would be right here, and you can see the nitrogen right there. Both DNA and RNA are formed when the sugar of one nucleotide bonds with the phosphate group of another nucleotide, and we call that the sugar phosphate backbone. So again, the phosphate and the sugar, the phosphate of one nucleic acid or nucleotide and the sugar of another nucleotide bond together in order to form that chain. But let's look at nitrogenous bases a little bit more carefully. So nucleic acids contain several types of nitrogenous bases. The sugar and phosphate group in a molecule of DNA are identical to the one from one nucleotide to the next. So every single nucleotide has the same sugar and phosphate group. In DNA, the sugar is always deoxyribose, and the phosphate group always contains the phosphorus and the oxygen atoms. The same holds true for the molecule in RNA, except that the sugar, instead of deoxyribose in DNA, every nucleotide in RNA has the sugar ribose. The one part that does differ is the nitrogenous bases, and that they differ from one nucleotide to the next. So DNA has 4 different nitrogenous bases. They have adenine, thymine, cytosine, and guanine. So A, T, C, and G. Chemically, they are classified into 2 groups. So A and G, adenine and guanine, are called purines. And these bases have 2 rings of carbon and nitrogen. Thymine and cytosine are classified as pyrimidines, which is a different type of nitrogenous base, and these bases only have 1 ring of carbon and nitrogen. So DNA is a double-stranded molecule that's in the shape of a twisted ladder. DNA contains the genetic blueprints that your body needs for growth and development. So for example, if you get a scratch, you need to replace those damaged cells with new cells, and so your DNA in your cell needs to divide as well, and that's a process called replication. A DNA molecule forms when 2 strands of nucleotides link together. So this is actually made up of 2 strands. They link together and twist around each other. This structure, again, is called that double helix. And the way that the 2 strands come together is based on the nitrogenous bases. So they pair up in a very specific way. So as you can see here, A always binds with T, and G always binds with C. Not only are the nitrogenous bases located inside the double helix, but they're also always paired up in this specific way. So again, adenine and thymine are going to pair up with one another, and cytosine and guanine are always going to pair together. This is what we call complementary base pairing. So just like peanut butter and jelly go together, adenine and thymine go together, and guanine and cytosine go together. So we know which bases actually pair up with each other. We know that A and T pair up and G and C pair up, but how exactly do they pair up with one another? Hydrogen bonds actually form between these bases in a DNA molecule. So remember how the nitrogenous bases adenine and guanine consist of 2 rings of carbon and nitrogen called purines, while cytosine and thymine each contain only 1 because they're pyrimidines. Inside the double helix, a double ring structure always pairs with a single ring structure. So again, the adenine and guanine, right, have 2 rings, whereas the thymine and cytosine only have 1. So the double ring structure is always going to pair with another single ring structure. Between the 2 bases, we have hydrogen bonds that form. 2 bonds occur between the adenine and thymine, and 3 bonds occur between cytosine and guanine. These bonds give additional support to the molecule to make sure that it keeps its shape. You may think of the bonds between the bases in the DNA molecule as rungs of a ladder. So if this was a ladder, the bonds would be this portion right here, whereas the outer rails are formed by the sugar phosphate backbone. because bases bond in a very specific way, if you know one side, so if I were to give you just the left side, you could predict the sequence on the right side. By knowing the identity of one nitrogenous base in a molecule of DNA, you can predict the identity of its pair in the other strand. So in other words, the sequence of one strand determines the sequence of another. The sequence of bases in a DNA molecule contains the information that organisms need to build proteins and carry out many important life processes. So we talked about the structure of DNA and the different bases that are present in DNA. Now we'll go ahead and talk about RNA. So RNA, unlike DNA, is actually single-stranded. RNA and DNA are similar in some ways, but there are some differences that are very important. For example, the sugar phosphate backbone of RNA is made up of the sugar ribose, and that's why we call it RNA instead of deoxyribose, which is in DNA. RNA also uses a nitrogenous base called uracil instead of thymine. So instead of thymine, it's going to use uracil, so that means that adenine is going to be bound to uracil instead of thymine. And finally, RNA exists most of the time as a single-stranded molecule, whereas DNA is typically a double-stranded. While DNA contains the genetic instructions for making proteins in the sequence of its nucleic acids, RNA helps cells read those instructions in DNA and uses them to make proteins. The goal of RNA is to take the instructions of DNA to the different organelles in the cell to help make the proteins. And there are actually different types of RNA that are involved in making proteins. So RNA plays several different roles in translating the information that was once in DNA so that we can string together amino acids to make proteins. So there's mRNA, which is the messenger RNA. We have ribosomal RNA, which is the rRNA, and we have transfer RNA, which is the tRNA. So each type of RNA is produced during a process called transcription. So transcription is when we use DNA to make RNA. So each of these are made during transcription. RNA plays the role of an intermediary between the information in a strand of DNA and the protein produced from that information. So basically, we use RNA as the intermediary structure between DNA and protein. And we'll be going over the specifics of the central dogma of biology, which is how we go from DNA to RNA to protein, in a later unit. So the summary of what we went over today is we went over two different types of nucleic acids, RNA and DNA, and we looked at its structure, how it's made up of 5 carbon sugars. For RNA, it's ribose. For DNA, it's deoxyribose. They both have phosphate groups and they both have nitrogenous bases, except RNA has uracil and DNA has thymine. The information in DNA is encoded in the precise sequence of nitrogenous bases in its structure. RNA uses those sequence of those bases as a template so that it can help make those proteins. So again, DNA has information, RNA uses that information to help make proteins. The information in DNA is passed from generation to generation. The last difference between RNA and DNA that we talked about as well is that DNA is double-stranded, whereas RNA is single-stranded. Okay, so some important vocabulary that we went over is DNA, RNA, a nucleotide, which is the subunit of a nucleic acid, nitrogenous, which means it contains nitrogen, and we use that word to describe nitrogenous bases, deoxyribose, which is the sugar in DNA, ribose, which is the sugar in RNA. We have the different nitrogenous bases, which is adenine, guanine, thymine, and cytosine. These are all found in DNA. And then we have uracil, which is found in RNA instead of thymine.